Kia ora tato. Welcome to the Genomics Aotearoa seminar. And today it's a pleasure to welcome Shujun Ou from Ohio State University. So I'm quite excited by this talk. We often, in the past, we've been looking at transposable elements as something that's very difficult to deal with and put them aside as a bit of a too hard basket type of genomic features that we don't really look too often, but they're super important. The more and more we look at them, the more we realize that they're critical for in controlling a lot of phenotypes. And so that's why it's really important to develop some robust bioinformatic tools that can deal with transposable elements. So Shujun is leading a team that's very prominent in that space. His record of tools that he developed and his team developed is quite impressive. So it's quite exciting to have you today, Shujun, presenting to us. So yes, let's hear what you got to tell us. Thank you for joining us.
Well, thank you, David, for your nice introduction. And thank you, Mick and Cecilia, for inviting me to this nationwide seminar series. It's humble me to present my work in here in this platform. So today I will talk about the differences in activity and stability that drive LTR retrotransposon accumulation in tropical versus temperate maize. So, like David mentioned, I'm an assistant professor at the Department of Molecular Genetics at Ohio State University. So I established my group 3 years ago. So we all know angiosperm genome size vary greatly. So you can tell in terms of genome size from prokaryotes to eukaryotes, and especially in flowering plants, the genome size vary in over 2,000 folds. With the smallest genome size is like less than 100 megabases, and the largest one over 140, 50 gigabases. So with this huge genome size, people are wondering what are the things stuffing into the genome, and that produces paradox called C-value paradox. So the genome you see from this very tiny genome strawberry, and for the genomes, this lily, Japanese lily, seemingly they have similar plants, right? But how come the genome size varies so much and their phenotypic complexity does not change that much? And if you look at the content of transposons, here I look at the LTR content, you can see it's almost linearly correlated with their genome size. And this was using genomes assembled like 7 years ago. That means they were scaffold and highly fragmented, and still you can see this very obvious trend. So that's one of the answers to the C-value paradox is the existence of transposable elements. Especially in plant, long-terminal repeat retrotransposon is dominating almost every plant genomes that I have seen so far. And as in maize, within one species, transposons also contribute to genome size variation. So here, researchers sample different maize in 2 locations in Mexico and the Andes, and in different elevations on the x-axis. And then they estimate the size of TEs from these genotypes, and they found out the negative correlation between TE content and the altitude. That means maize sampling from a higher altitude and cooler climates has smaller genome size and replicate in 2 geological locations. And these researchers, they collect some maize and self them in different generations, and then measure the genome size change across selfing generations. And they found at least 3 lineages, not all of them, had great reduction of genome size, all mostly due to transposable elements deletions. So not just genome size, they also contribute to biological function. So like TEs can insert into genes, right, and directly mutate your gene. Although this is rare, but although already we observe some important agronomic traits were contributed by this process. And also TE activity can duplicate genes that can change the expression level of a gene like this sun locus produce the gene and then resulting into this cell elongation being promoted. The thing that we most interested in my current lab is the regulatory impact of TE's insertion to gene expressions. So there are two consequences. TE insert nearby genes, which are like most likely not duplicating genes, not into genes, but most likely they would insert nearby genes. They can either activate or deactivate your gene depending on the mechanism driving this. So for example, this MIP1 gene is not expressed in apple, but expressed in grape, that resulting into two color of skins. And there's a TE, different TEs in different species insert in front of this MIP1 gene, resulting into the activation of the MIP1 gene in apple and the activation of the MIP1 gene in grapes, and this shift of color skins. And if you partially delete the TE, you end up in this intermediate form, like reddish, pinkish gray skins. So what I'm trying to answer with this project is how TE's content vary between one species, like tropical and temperate maize. Um, and if there are variations, what are the possibilities and what are the mechanisms driving such variation? So these two questions were difficult to answer previously because we didn't have high quality maize genome assemblies. And if we have several already, but those genomes were in uneven quality. That it's difficult to distinguish whether this is due to biology or technical artifact. And a chance come in a couple of years ago when we sequenced 26 diverse maize lineages. So the thick bar here are representing the collection of these lineages. So they are like representing the diversity of maize. And that includes 13 tropical maize lineages and 10 temperate maize lineages, including popcorns and sweet corns, and some other mixtures. So these maize lines are inbred lines that were used as a hybrid parents that cross with B73 as the reference to create a population called nest association mapping population for quantitative genetics and also functional genetics, and which is actually important lines that has been sequenced whole genome. And the genome was sequenced in long reads and with consistent high quality so that we can rule out the technical artifacts. And then we can focus on the biological differences and finding the contribution of TEs. So to study TEs, you have to annotate them, right? That's how I developed a method called EDTA to annotate these maize genomes. It was developed based on benchmarking. So you can read this paper. We use the rice genome as the platform because rice, we know all the annotations by curation. And then after benchmarking, we collect the high quality tools and then combine them with lots of filtering to further filter down the remaining noises and generate a collection of TE sequences. We call it a library. And then we use the library to make homology annotations. However, this is a method developed for a single genome annotation. Now we have 26. So I just extend the method in this study to first apply it in each genome and then process the library to collect those high quality multi-copy sequences to form a non-redundant pan-genome library. This component is called pan-EDTA. So this pan-genome library is again supplied to EDTA to annotate the genome again, so that you have a consistent annotation of TE families across the genomes. So that allow us to compare the variation between different individuals. So this is the pan-genome annotation of TEs in maize. You can see, just looking at colors, the colors are quite consistent. That means we are doing a good job here that providing this consistent annotation across maize genomes. And the coloring scheme here, green is the tropical maize, and the other color except gray are temperate maize, and the grays are intermediate genotypes. So a little bit more about the transposon biology. So based on the way they duplicate, we can classify them into two major classes. One is through cutting the entire double-strand DNA and then move to a new location. We call it cut and paste mechanism. This is called the DNA transposon. And the other mechanism is you can transcribe into an mRNA and then use the mRNA to reverse transcribe back to a DNA and then integrate into a new place. That is called the RNA-mediated transposons or retrotransposons, or the copy and paste mechanism. And we can further classify DNA transposons based on their presence of the transposase or the protein machineries that facilitate their uh, mobility. With the machinery, we call it autonomous elements. Without the machinery, we call it non-autonomous, that the element need to have other machineries to supply the movement, not by themselves. And if you further fragmented them, you can become highly fragmented pieces or remnants. So the class 1 retrotransposon include LTR with a long terminal repeat and the non-LTR that without the long terminal repeat, usually cyan lines. And in plants, as I mentioned, LTRs are a lot, and in human, animals, vertebrates, lots of them are lines and signs. Okay, so today the focus is on LTRs. So let's look at the size of the family as we can compare across individuals. We first interested in like how varying are the size of the family. We found out, so first we put them into three classes. Rare are the one that are a single copy, and the size less than 100 kilobases is actually not a lot, and the size larger than 100 megabases is even less. Only 0.2% of the families have 100 kb across the whole genome. So this is in number, but if you put them into base pairs, it's a reverse order. These large family, they are dominating the whole genome, and those unique ones are just like a tiny fraction. And if you think about like how are these family how are these families being shared or unique between individual plants or genotypes? So we do this saturation test. So with the x-axis being the number of genomes randomly add to the pool, and then the y-axis being the number of family T families that can be found in this subset of the genome. And the second y-axis is the percentage of families. So you can tell as far as like 10 genomes added into the pool, you can find over 95% of the T family, which is the majority, right? So basically, the variation between individuals of the same species in maize is not that big. And if you rank them from the largest family to the smallest, and here is the 100 largest family in maize genome, the largest one is almost 80 megabases, two-thirds of the Arabidopsis genome for only one family. So this is huge. And quickly drop into around like 10, 15 megabases families I think this is the 50 largest. Yeah. And then contribute over 50% of the genome. And the error bar here is the variation between the 26 maize genomes. And you notice most of them are not that varying, right? They have little variables. That means they're not changing between individuals. And some have some variation, like this one. So if we put the family size variation as a variable and then compute a PCA, a principal component analysis, we can actually see the family size variation can classify our genome into two major groups. So tropical in green and then temperate in non-gray colors. So that suggests some divergence between tropical and temperate maize genomes. So we further identify those families with the highest variation in family size, and then put them into context of temperate and tropical genomes. And we found out those highly varying families are generally larger in tropical maize genomes. It's very significant. So with this, we further um, construct a very simple number using the family size average in tropical minus the family size average in temperate to see how much is the variation. So we've put them into three categories. Families larger in temperate genomes, basically no difference, and family larger in tropical genomes. So you see more color in this category. That means families are generally larger in tropical maize genomes, and there are like 145 of them, larger than 25 kilobases on average. So this is not a huge number. And if we put this in a further context, in a different classification, so the blue color here is the LTR, and line is here. Helicon is the DNA transposon, and orange is also DNA transposon. And beyond zero means families are larger in tropical. Below zero means families are larger in temperate. And as you see, most of the differences are contributed by LTRs, with some contributed by helicon and maybe capta. So that further encourage us to look at LTR, and this number is more than the contribution of the LTR itself in the genome content. LTR is like 75% of the genome, and this contribution is over 90% if I remember correctly. So this is more than its random contribution. So together, LTR retrotransposons contribute over 40 megabases difference between tropical and temperate maize genomes, with some from others. So we wonder why, right? There could be two possibilities. So one is like the LTRs are more actively amplified in tropical maize that make it larger. And the second hypothesis is the LTRs in the temperate genomes are being more removed, downsizing. That you don't need to do anything in the tropical genome, that the temperate genome just being smaller. So the two hypotheses are very different, but can result into the same observation. So to distinguish these two, we need to tell the dynamic of these LTRs, like whether they amplify or whether they are removed. So the removal of LTR can be quantified. So here is the structure of the LTR with the 5' and 3' long terminal repeats. And because of the sequence similarity, there is a process called illegitimate recombination that you can have this local recombination resulting into the deletion of the majority of the LTR and then the remaining of one LTR region called solo LTR. So if you put this into the same family, and then you calculate the number of solo LTR over the number of intact LTR, you can kind of have an idea on how intense this family is being removed, with a higher ratio being more intense removal. So that is a reflection of the removal of the family. Then we start to distinguish removal and amplification dynamics by first comparing whether the family size are different. Of course, the majority of them are not different, but some are different. And if they're different, we ask whether their solo intact ratio are statistically different between temperate and tropical maize genomes. We have a sample size of 10 and 12, so I think that will give us enough statistical power to tell the difference. And if the removal dynamic is significantly different, we can tell this family is a removal family, is a family under the removal, driving by the removal dynamic. And if there's no difference, but the family size is significantly different, that we can tell this family is driven, the different size are driven by the amplification dynamic. And of course, we have the things that are not different, and that can be either balance, removal, or random drifting. We have some small observations here. So the major focus is on the removal and amplification. Um, By looking at the family that are larger in tropical genomes, we can further put them into the four quadrants, which is tropical amplification. So this amplification can be amplified in tropical or amplified in temperate, right? But if we look at the genomes that are larger in tropical, that we can tell this is amplified in tropical, and also removal in temperate, making it larger in tropical genomes. And similarly, there are some temperate larger families and temperate amplification and tropical removal, but they are generally smaller. Each dot here is a family, and the dot size is the differences between tropical temperate average differences. And you see the dots are generally smaller here, and we have lots of random drifting. So the larger dots here are the tropical amplification and the temperate removal families. But we wonder, when did these kind of things happen? So we can put this into a time context by look at the structure of LTR again. Upon the insertion of the LTR, like when they are newborn, the 5' and 3' LTR are identical due to the reverse transcription, and that they start to function as a molecular clock that they can record their own age by, and then you can tell how old they are by comparing the 5' and 3' LTR mutations. So we calculate the age of each LTR, and then we look at each family, their age distribution. We can see three patterns. So this pattern is we call them young family. That this family has the most of the LTR are very young, almost zero age, and then a moderate family is like there are some very young families, some very young individuals, but the peak was older than zero, and the old families is like they don't generate many young LTRs. Basically, they are not active anymore, and their peak was past in the past tense. So we classified in the three groups, and also look at the age distribution of the top uh, family. This is the largest families, and they happen to be all LTRs because LTRs are very, very long. In maize, and average LTR is like 9 kb. So with this size and the copy number, they easily dominate the genome and being the largest families. And if you rank them from young to old, you see this trend. And basically, it's like different waves. And these waves were older, these waves younger, 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 younger. And this is like very recent and the youngest. And the old ones are kind of like around half of the large families. This suggests the past activity. And these moderate and young families are suggesting the recent dynamics of the TE. I didn't mention the clade. If you're interested, you can look at them, but the majority of them are sire and retent clade, which makes them very successful. And if you also want to put them into context, whether they are um, uh, tropical amplification, temperate removal, like if you just look at the green and the salmon color, you found them most greens are here, salmon are here, and the blues, these are random drifting, like these old ones are random drifting. So by just using two matrix, the solid intact ratio and the size differences with a t-test, we can classify LTR families into four categories. And then we found out both amplification and removal contribute to these family size differences, but the amplification seems contribute more to this dynamic. So that kind of answer both of our hypotheses were correct, but amplification is the driving mode. So we further put into three contexts of the age, young, moderate, and old, and into different LTR superfamilies, TY1, TY3, and family without any classification information. We call it unknown. And also put into the dynamics of the four dynamics. Salmon is removal, green is amplification. So you can tell the young ones contribute a lot. Oh, the y-axis is the accumulation size of the tropical maize differences. And each box here is one family. So you see several families driving up the size differences, accumulating 15 megabases. You remember total is like 40 difference. And mostly are TY3 that are mainly enriched in centromeric regions that are gene rare and low recombination. So in those moderately old, the contribution coming from amplification families, still some, but not a lot. And for the old ones, the contributions start coming from these blue random drifting families. So it's very interesting. Like the older they are, the more drifting they are experiencing. So what are the mechanisms, like the molecular mechanisms driving these differences? So we look at the expression and the potential of expression itself. So Jonathan Gant from Georgia, University of Georgia, so he analyzed the methylation data from enzymatic mesoseq, so EM-seq, um, and identify those unmethylated regions. And I further put them into context of LTR, like are these UMRs coming from the LTR itself, like enclosed within the LTR? So, and then identify those LTRs carrying unmethylated regions that potentially can be expressed. So here is just one example that we know this centromeric retrotransposon repeat is called CRM2. We know they are active, and we also observe lots of UMRs coming from the CRM2 copies. So these are the UMRs. So we also, with them, we have confidence to look at whole genome, and we separate them based on the superfamily and also based on their location with genes. Some of the TE are located in the intron of genes, some are in the genetic regions. So it seems like genes are not driving this pattern, right? Because outside of genes, we still see the TEs, and the length is around 300 um, uh, nucleotides, similar in TY3. There's a pic here. We also have this accessible chromatin region data coming from ataxic data. It's also a whole genome level. So we also look at accessible chromatin is also around the same region, so around the same length of this ataxic and the UMRs data. And this happened to be around 2 to 3 nucleosome in length. We didn't check, or we didn't have the power to check whether there are any nucleosome occupancy in these UMR or ACRs, but they happen to be around 2 nucleosomes length. So potentially they are um, releasing 2 nucleosomes to allow the chromatin being accessible and being methylated in LTRs. But are, are, are these elements carrying ACRs and UMRs has any relevance to the data that we observed? So yes, this is UMR on the left and the ACR on the right. And the same context, TY3 superfamily, TY1 superfamily, young, old, and moderate dynamics. And the y-axis here are the number of families that we can identify. And the color here are the different dynamics. And I put them into 2 columns. I count them in tropical genomes on the left and temperate genomes on the right. So you can see the dominating color is green, which are tropical amplification families. That means these amplifying families, they have more UMRs and ACRs than other dynamics. And then secondly, if you look at tropical and temperate differences, a lot of them are coming from more numbers in tropical maize genomes than temperate, especially if you look at the ACR here, it's more dramatic. So that means these tropical amplifying family are more open chromatin and methylated in tropical maize genomes. And they are mostly coming from young and moderate families. So all support each other. So we wonder this open chromatin or accessible chromatin, do they actually change the expression? right? So we look at LTR expressions. So Arun and Sarah from our state helped with this analysis. From the genomic project, we sequenced a lot of libraries for gene annotation purposes. So these libraries cover 10 tissues per genome across the developmental stage of maize, like from seed, embryo to vegetative growth, leaf, and flower and tassels. And each of the tissue with biological replications, and each of the genome was sequenced in the same level of depth. So we have hundreds of libraries, but these are all Illumina, so that give us a, a, a challenge, technical challenge, because short reads aligning to repeat is basically challenging or not possible in many locations. So instead of focusing on the expression level of a single TE, we focus on the expression of the whole family, so that even the alignment is not accurate on that individual, we count multiple alignments and then evaluate the expression level of the whole family altogether and see if any families are differentially expressed between temperate and tropical maize. So we make the contrast and identify those differentially expressed T families and put them into context again, and we found them explaining almost 60% of the T content variation. Like those differentially expressed T families, they contribute to 60% of the tropical temperate T family size differences. And they're mostly coming from young TY3 families. So again, this aligns with our previous observation on the ACR and UMRs that they're coming from young LTR TY3 superfamilies that are higher expressed in tropical maize genomes. So these are high expressed in tropical maize, not in temperate maize. Right. So I was to push. I tried to push the limit by further look into population genomics. We have a population of 26. It's not huge, but still is a small population. Can we tell the dynamics of individual LTRs in this population? So to identify polymorphic LTRs between maize genomes, which is super challenging, um, I use this micro homology, considering the flanking region, and then identify if this LTR is shared between two genomes or unique in just one genome. So that I can know the presence and absence variation of each Ts across the 26 maize. Well, this is very hugely simplified. We have 50,000 of LTRs and then 26 maize genomes. We need to calculate the pairwise presence and absence variation, lots of compute, and then condense into this diagram. This is the insertion frequency counting from 0 or from 1 to 10. And I dropped the number of tropical to 10 to match the temperate maize genomes number. And the y-axis is the frequency of the proportion of TEs being in such frequency. So of course, most insertions are rare. So changing from here, this is one insertion and this is two insertion. And if the insertion present in 10 genomes, this is 10 insertion. That's what you see here. So most of the LTRs are rare and some are all shared. And we saw the difference between temperate and tropical maize. Why? So this can be explained by bottleneck effect because maize was domesticated in Mexico region in the tropical climate, and then some migrate up north and become temperate maize. And this migration itself is a bottleneck effect, and this bottleneck effect will result into the reduction of rare variants and then the increase of the middle frequency variant. That's exactly what we see. So we saw a reduction of rare LTR or unique LTRs in temperate maize and increase of the mid-frequency LTRs in temperate maize. So this can be explained by bottleneck effect. Um, and we also look at those unique TEs unique in temperate or unique in tropical, and we found they are younger than those shared. So that means other than this population dynamic effect that the itself do not need to do anything. They're just being lost by artificial bottleneck or migration. But some of them are still producing new copies through this process in both temperate and tropical maize, at least in tropical maize because it's younger. And temperate maize, some of them may be shrunk by population bottleneck. Some of them may be induced by new insertions. So we want to push the limit again and see if there's any biological relevance, like why tropical maize need to have more amplifications or need to have more insertions. Why? So one thing we look at, and we learned this from a collaborator, that they observe more NLR genes or more Ts surrounding NLR genes in Arabidopsis. We want to test its hypothesis and look at so Armin look at maize. So we first annotate all the NLRs from scratch and then put them into whether they are clustering or whether they are singletons, and then look at the context, whether the LTRs are more enriched in a singleton context or a cluster context. So we found a slight increase of frequency for the young LTR families in where? In the NLR clusters when you compare to uh, uh, NLR singleton. We don't have the data here. You compare NLR cluster to NLR singleton or non-NLR singleton or non-NLR cluster, you still see this enrichment, which means there are more enrichment in the NLR clusters. What are NLRs? NLRs are leucine-rich repeats that are mainly function as disease-resistant genes, R genes. So they have uh, this enrichment in the NLR genes, suggesting there's something going on between these two. And they're not enriched in the non-NLR gene clusters. Like other gene clusters, they don't have such enrichment. So which means the fast evolution of NR clusters may correlate with, is already correlated with the fast amplification of LTRs, but we don't know which one is driving which. It can be NLR driving LTR or LTR driving NLR, but we haven't dig further. So we haven't talked about this removal yet, right? So I mentioned removal is due to this local recombination, but we look at the sexual recombination and we found some correlations. So these temperate removal LTR families are more found in genomic regions that have higher sexual recombination rate. So the illegitimate recombination, as far as I know, is not driven by sexual recombination, but there are some shared physical locations between the two things. So I want to summarize the results. So first, we identify LTR retrotransposons contribute a lot to the maize genome size differentiation. Well, I need to correct myself, not a lot, 40 megabases, and it's not driving the maize genome size difference. The driver is heterochromatin knobs. Those are tandem repeats. But still, we found like T contribute around 10% of the genome size variation in maize. And we found both LTR amplification and removal shape the maize genome size in the T content. And then the young elements contribute most of the content variation. And the tropical amplifying LTR families were more unmethylated, chromatin open, and high expressed that may have been driving their activity. And the reason why they are more active is likely due to disease, this R gene arms race. And we also observe a smaller genome size in temperate genomes, likely contributed by either removal. The removal dynamic can be solo intake ratio, and also some may contribute by this bottleneck effect. I went over this, and with that, I want to thank my postdoc advisors. So this is my last postdoc project. Matt Huffer at Iowa State, Candy Hirsch at University of Minnesota, my co-advisors, and my collaborators in different, like in Johns Hopkins, Cold Spring Harbor, Minnesota, Georgia, and Galaxy. And now I have my own lab. So I have a privilege to work with all these genius young people and keep asking what's the consequence of T activities? Like, why do they active? Like, I want to look at the positive consequences, right? If you want to find negative, you always find negativity, but I want to be positive. So I want to see like how T's may contribute to biological function, regulation, and evolution. So this, hopefully I can share more result in the future. And with that, I would like to take any questions you may have. Thank you so much for your attention.
Thank you, Shujun, for joining us. And it's never easy to actually give an online seminar to a bunch of people on the other side of the world. So thank you very much. Really impressed by your talk and your work. So thank you.
My pleasure. Thank you for inviting me again.